I am Mariam Ram of TNQ. Welcome to the third and final seminar of the TNQ-Janelia India COVID-19 series. To bring this series to a close, we are privileged to have, as our speakers, Professor Benjamin Tennover and Professor Florian Krammer of the Icahn School of Medicine, Mount Sinai, New York. In presenting these seminars, TNQ has partnered with the Howard Hughes Medical Institute's Janelia Research Campus in Ashburn, Virginia, which has provided the scientific and technical expertise and has been supported by NCBS, the National Center of the Biological Sciences in Bangalore. For this, I thank Professor Ron Vale, Executive Director of Janelia Research Campus, and Professor Satyajit Mayer, Director of NCBS. Janine Stevens and Sharda Vishwanathan of Janelia identified the speakers and put all the programs together. For the seamless presentation of these seminars, I applaud Matt Staley, John Rapole, and Alex Fadeyev, also of Janelia. They have been ably supported by our TNQ team. Thank you. I now hand over to Ron Vale.
Thank you very much, Mariam. And again, I also want to thank you for the wonderful work that you've been doing throughout the years on science communication in India. Certainly good, accurate communication of scientific topics is more important than ever on all topics in science, but especially on COVID-19. So Janelia is extremely pleased to be able to partner with TNQ in this lecture series. And also, I've personally been very interested and enjoyed the last two lectures and looking forward to today's lectures on the very important topics of immunity and vaccines, which everyone is very interested in. And I would like to echo Mariam's thanks to all the Janelia organizers. You'll hear more from Sarada and Janine later as they will organize the question and answers. So now I'd like to pass things over to the director of the National Center for Biological Sciences, my colleague, Jitu Mayer.
Well, thanks, Ron, and good morning, good evening, and good afternoon, everyone. At a time when the COVID-19 crisis is raging and India is one of the epicenters of infection, there has been an unprecedented collaboration amongst scientists near and far to understand the biology of the virus, study its mechanisms of spread, and also its amelioration. And the National Center for Biological Sciences in Bangalore is delighted to support this activity and collaborate with TNQ and Janelia to bring you today's lecture on the amelioration of the disease. And Sharada is going to introduce the speakers in a short while, and before she does, I'd like to introduce you to our panelists from India. We have Dr. Gagandeep Kang, who is a clinician scientist, an outstanding clinician scientist from the Christian Medical College, CMC in Vellore, where she's professor of microbiology. Until recently, Dr. Kang was the director of the Health Sciences Translational Institute in Delhi, and then now she's come back to CMC in Vellore. She also happens to be the first woman in India to be elected as a fellow of the Royal Society. This is for her work on enteric infections in children, particularly on the transmission and on its transmission and immune responses. Her current studies include active hospital and community-based surveillance and clinical trials of new and existing vaccines, a central feature of today's talk. And then we have Dr. C.S. Pramesh. He's the director of the Tata Memorial Hospital and professor and head of thoracic surgery at the Tata Memorial Center, both in Bombay. Dr. Pramesh has strong interests in clinical trial designs, surgical trials, and therefore he is deeply interested in understanding and is keeping track of how effective drugs for this pandemic can be made available to the Indian population and for the population at large. Finally, our moderator for the panel is Professor Aurnab Ghose, a cellular neurobiologist at the Indian Institute of Science, Education and Research in Pune. And during this ongoing pandemic, he retooled himself to help establish a COVID-19 testing center and is also a lead investigator in Pune city's first serological survey, where he's currently investigating the humoral immune response to the SARS-CoV-2 infection in communities in Pune. So without further ado, let me invite Sharda to take over.
Thank you, Professor Mayer, and welcome everyone. Today's talks will be followed by an expert panel discussion and then an audience Q&A. So please submit your questions at any time during the talk, and please do mention who your question is for. With that, on behalf of TNQ, Janelia, NCBS, and all of us here, it's my pleasure to introduce the speakers for the day. Our first speaker is Professor Benjamin Tennover. Professor Tennover obtained his PhD in medicine from McGill University, followed by a postdoctoral work at Harvard University. He is currently a Fishberg Professor of Medicine, an Icahn Professor of Microbiology, and the Director of the Virus Engineering Center for Therapeutics and Research at Mount Sinai. The Tennover Lab studies at the molecular scale how viruses interact with the cell during an infection. And using these approaches, Professor Tennover has made some significant advancement in our understanding of the systemic immune response to SARS-CoV-2 infection. We'll hear all about that in his talk today in a bit. Our second speaker is Professor Florian Krammer. Professor Krammer obtained his PhD from the University of Natural Resources and Life Sciences, Vienna, followed by postdoctoral studies at the Icahn School of Medicine at Mount Sinai. He's currently a professor of vaccinology, also at the Icahn School of Medicine, and his work primarily focuses on understanding the molecular interactions between the virus and the antibody against this virus, and then translating this knowledge into novel, broadly protective vaccines and therapeutics. So stay tuned to hear more about all that. Professor Krammer and his team have also made some remarkable contributions to this field, including serological assay for the detection of SARS-CoV-2. So once again, Professor Krammer, Professor Tennover, welcome. Thanks for your time, and we look forward to your talks.
Great, thank you so much. It's great to be here. Let me just share my screen here. So I will repeat that thank you to Janelia and to TNQ for organizing this and making it all so seamless. I hope everybody can hear me. I wish everybody a good morning, good afternoon, and good evening as well, depending on where you are in the world. So today I'm going to present a mix of what my lab has been doing since March. It's a combination of some published data, some data that will soon be published, as well as some unpublished data that's still relatively new to the lab, but I feel strong enough in the conclusions that we can discuss it. Please feel free to throw questions on the chat, otherwise we can discuss later at the end. All right, so I don't think anybody in this audience is unaware that there is a pandemic going on that is really unprecedented in proportions, at least since the last 100 years. And we're definitely going to discuss a lot about this. I'm going to focus more on the molecular biology of the virus, and I'm going to assume that Florian will talk more about vaccine development. Just so that we're all on the same page here. This is what the virus itself looks like with regards to its assembled products. Just as a quick lesson, it comes in essentially as an infectious mRNA. So it comes in as a positive sense mRNA, which means that initially the ribosome can load immediately onto this incoming nucleic acid and start translating the open reading frame 1A and the open reading frame 1B, which is caused by a small little frame shift here. And this is going to create the necessary machinery required for this virus to amplify this piece of RNA in both orientations. And that really comes in the form of these subgenomic RNAs here that you see to the right. And so the way this happens is after ORF1A and ORF1B are made, the virus begins making what almost looks like replication. So it starts making the opposite sense of this piece of RNA going this way. And the virus does something quite unique in that it has these transcriptional regulatory sequences which occur periodically along this subgenomic piece here beyond 21,000 nucleotides, and the polymerase then jumps to the very end of what is the 5' end of the incoming genome to make the end of the subgenomic RNA. And it does this so that each subgenomic RNA can be recognized by the polymerase. But I mention it because it actually is quite a messy means of transcribing and replicating your virus. And as a result, you produce a lot of RNA pieces that are bound to the antisense RNA, producing what we refer to as double-stranded RNA. So here what you're looking at here's an uninfected plate of cells where the nuclei are just stained blue. So those are just all the nuclei here. And if you infect this with SARS-CoV-2, the virus that's causing COVID, at a multiplicity of infection of 1, so that means one virus per cell, and you let the infection go for an hour, and then what we do is we stain with an antibody that recognizes the production of double-stranded RNA, and that's what you see in green. And all I really want you to appreciate from this slide is how messy this virus really is, and this is going to, I hope, resonate throughout this talk as to the consequences of this essentially sloppy replication that the virus uses to produce these subgenomic RNAs. So my lab does a lot of sequencing. So this is where you sequence everything in parallel and you get a snapshot of how the cell is reacting at any given time and what the virus is doing at any given time. And so what you're looking at here is essentially a histogram. So these blue lines along the bottom here represent the number of captured reads that align along this genome of almost 30,000 base pairs, which is 3 times larger than any other RNA virus that we know of. And it's quite a unique family of viruses, the Coronavirinae, in this way. So the blue is what happens if you infect a human cell line called A549. It's just a cell line, it's a tool, it's not a particularly good cell line, but it's a useful thing because we can manipulate A549 cells and therefore we can dig deeper into the molecular biology of this virus. But A549 cells lack the receptor, the ACE2 receptor, and so without the receptor you can appreciate, I hope, that there is very little viral reads that ever get into the cell. And this is probably from some periodic ACE2 expression that allows a few viruses to get in and generate these reads. However, if you just simply add ACE2 to these cells, you can see that you go up many, many orders of magnitude, and now you're achieving anywhere from 1,000 to 10,000 reads, meaning individually captured sequences for any one given genomic position on the genome. And again, here what I really want you to appreciate, though, is that with this asterisk here, this lighter red color, even if you come in with an MOI of 0.2, meaning 1 virus for every 5 cells, or you come in with an MOI of 2, meaning 2 viruses for every cell, at the end of the day, the RNA in the cell is saturated, meaning the virus is raised to the same level regardless of where you start. And this really doesn't have to do with anything other than the fact that this is essentially the maximum amount of RNA that the virus can usurp and the amount of tools that the virus can usurp in these cells. And just to give you a better sense of that, if you take a look at, so you take a snapshot of this sequencing, right? So the total RNA that the cell is making in response to the virus and the amount of RNA that the virus is making to replicate itself. And so I hope you can appreciate here is at 6 hours, if you take a snapshot of that RNA, maybe 1% of that RNA is virus in origin, and the other 99% is of viral origin. At 12 hours, it goes to maybe 5%. By 18 hours, we're already over 20%. And at 24 hours, we can achieve almost 80%, meaning 80% of the RNA in an infected cell is of virus origin, which is an incredible feat for the virus. And it really can only be achieved by the fact that the virus is not only destroying the host RNA, but then, of course, also making its own RNA. So it really is completely taking over the cell. And if that was ever in doubt, this is some electron microscopy work that came out of my lab. And you can certainly appreciate just from a bird's eye view of what's happening here. So this is the nucleus of an A549 ACE2 cell, and this is what the cytoplasm really should look like here. However, if you just give it 24 hours of infection, it's not maybe surprising that we have 80% amount of RNA because you can see all these vacuoles that have formed, and these are all replication factories where this virus is simply propagating and making thousands and thousands of copies of itself, all at the expense, of course, of the cell. Moreover than that, if you sequence this, so these are volcano plots. And so what you're looking at is every dot represents a gene that is statistically upregulated. And so the higher it goes on the plot, the higher the p-value is, and the further to the right it goes or the left it goes, the more upregulated or downregulated it is. And the genes themselves don't really matter. All I really want you to get a sense of is how these cells are responding. So if you just look at the top, this is an MOI of 0.01, so that's 1 virus for every 10 cells. And you can appreciate that 8 hours, there's very little happening, and very few cells are infected. But very rapidly, you can see that the cell is just doing everything it possibly can do to try and get rid of this virus. And you can appreciate that as your MOIs get higher, that robust response seen here in all these red dots happens earlier and earlier and earlier because essentially it is the response that you're seeing, all these genes that are being induced, are a direct product of the production of that double-stranded RNA that I showed earlier, the sloppiness of virus replication. Now, what the cell is trying to do is detect that kind of PAMP, or that pathogen-associated molecular pattern. So here depicted in these red lines is one of those PAMPs with regards to SARS-CoV-2, and that's the production of double-stranded RNA. And so in our cells, we have lots of sentinel proteins that recognize things like that, like double-stranded RNA, pattern recognition receptors. And they in turn oligomerize, they activate a number of kinases and ubiquitin ligases that then activate a number of transcription factors that I don't have time to get into today in a 25-minute talk. But ultimately, what I want to convey to you is that the cells that are infected at that first site of infection in somewhere in the respiratory tract are really trying to do these two goals. They're trying to essentially induce a call to arms. So this is the interferon response. So it's going to release a family of proteins into the cellular milieu, into the environment, the surrounding environment. And the objective of this call to arms is to tell the neighboring cells that maybe aren't infected yet to fortify your defenses and get ready for this virus. And so they upregulate a number of genes that are designed to do a wide variety of things, from block transcription, block translation, stop transport. There are direct antiviral effectors, there are nucleases, all kinds of armament to slow virus infection. In addition to that response, those same infected cells are also releasing a call for reinforcements. And they're doing this in a very similar way. They're releasing proteins into the extracellular milieu. However, in this particular case, it's not interferon, but they're chemokines, which are, it's a fancy word for chemoattractive cytokines, or secreted proteins that are going to create a gradient to bring the adaptive immune response to the site of infection. And you need this because that's a far more sophisticated response where antigen-presenting cells can capture foreign protein and then make an antibody response to it. And so one of the things that came out really early, this is from a publication back in March from my group, was that if you come in with a low MOI, meaning that the virus has the capacity to follow its stepwise replication capacity and the way it's been evolved to do, what you find is that this blue box here that I've outlined here, these are the interferons. So the call to arms is completely lacking when you come in with a low multiplicity of infection. And because these cells are not having the opportunity to release those interferons and to induce that call to arms, all the effector genes here on the left are all white or very, very dimmed, which is unfortunate because that's what we use to slow virus infection at the site of infection to provide time for the adaptive response to come in. In contrast, though, if you come in with a high multiplicity of infection, meaning that you're going to make a lot of that double-stranded RNA very quickly without any opportunity for the virus to block that response, then you can actually see those that call to arms come back. You see all the effector genes come on, seen by here in red. So that's the first thing, is that the virus has the capacity to interfere and block the call to arms. Now, that in and of itself is not entirely unusual. Most viruses that cause disease, influenza amongst them, have the capacity to block the interferon response in some regard. What makes this virus particularly unique, though, is seen here on the right. So here on the right are all the cytokines, and this is the call for reinforcements in this analogy that I keep making. And what I want you to appreciate is that regardless of whether the multiplicity of infection is low or high, in both cases, you're getting this call for reinforcements. So this virus is blocking interferon, but allowing a lot of chemokine expression for the duration of the experiment. And so if we look at this by volcano plot again, what I hope you can appreciate is that at a low MOI, you're going to see that these chemokines here, CCL2, interleukin 6 is the pro-inflammatory cytokine, CXCL5, CXCL1, these are all genes that are involved in recruiting many of the adaptive immune cells to the respiratory tract. And again, if you come in with a high MOI, you still see all of those guys, but now you can add in the interferon members and that call to arm. And so there's really a big difference between these two models of infection. But of course, a low multiplicity of infection is more representative of what it would be like to actually contract the virus in the real world. And we can also see this exact same pattern. This is from COVID-19 patients, or postmortems actually, from the lung. And so again, we see a mixture of a few interferon-stimulated genes. So there is some interferon response, but really it is dominated by a high production of chemokines from the get-go. This is, again, just kind of reiterating the same point that if you look to serum in COVID patients, we see high elevated levels of cytokines and chemokines. And this is, I think, well established now in the field. And so this really comes out of what I was mentioning, the fact that this virus comes with many antagonists, including ORF3b, which has already been published and described by the Kato group, or the Sato group in Japan, as well as the PLP protease, which is thought to cut IRF3. And so we block half of the transcription factors, which is enough to stop the call to arms. However, there's this other set of transcription factors called NF-kappa B, which appear to be left on and unchecked in response to virus infection. And as a result, you're getting a lot of production of chemokines in the absence of this. And it's really this imbalance that's causing a problem. And so this was just the graphical abstract kind of summarizing that initial finding that, in contrast to the normal balance of chemokines and interferon that you might see with a respiratory virus, in the response to SARS, we see too much chemokines and not enough interferon-stimulated genes. All right, so what I really want to talk to you today, though, is an animal model and a move towards, it's great that you can learn all of this biology about the virus, but can you actually utilize that in any way to in fact, do something useful and therapeutic against this virus? And so we initially started with a ferret model, but the ferret model is limited in the fact that the ferret model, we don't see a lot of lower respiratory infection. It's really contained in the upper turbinates. So in contrast, the hamster does develop much more pathology with regards to the respiratory tract. And so we've done this by transmission. So if you simply, by fomite, if you simply take dirty cage material and put it into a naive cage of hamsters, those hamsters will all get SARS-CoV-2 as a result of the virus hanging around on particulates in that cage. You can take an infected hamster and put it into a cage with a naive hamster, and 100% of the time, it will spread that virus to the other hamster. You can put a tiny drop of virus on the eye of a hamster, and you can find robust replication in the lung. Or, of course, you can administer virus directly to the nose, and of course, that also induces a wide variety of infection. If you then take this intranasal route, so just simply direct infect, I also just want to show you how sensitive this is. And so again, if you come in with just 10 infectious units, 3 to 4 times in this particular experiment, the hamster comes down with a very high level of infection in the respiratory tract. At 100 PFU inoculation, you see all the hamsters get infected. And interestingly, at 1,000, 10,000, and 100,000 PFU, all the hamsters get infected, of course, but you actually see lower viral levels. And again, this goes back to what we were just showing you, that the higher that you come in, the more virus that you come in with, the more production of these PAMPs you produce. And it provides actually some time for the host to respond to those PAMPs before the virus has the chance to completely shut down that system. And this is going to become really relevant when I get to the very end of this talk. So just quickly to go through this, if you come in with hamsters at 100 PFU and you just simply watch the titers on any given day, so what you're looking at is day 1, day 2, day 4, 6, 8, and 14, you can see that at 100 PFU, the virus rapidly amplifies in the respiratory tract, and then the animal clears it. And you'll see in a moment that this is due to a nice antibody response. And 10 to the 5 as a model system also works. So this is a lot of virus. And so as a result, you don't have this nice peak. You basically just start high, you maintain high before you eventually come back down. And so for the majority of this talk, I'm going to always refer to low multiplicity of infections. The animals also suffer morbidity, so they don't really lose weight, rather they don't gain weight. So these hamsters are generally 4 to 6 weeks of age, and they eat a lot and they grow a lot. So when you get them, they increase in weight every day, very happily in their cages. But if you inflict SARS-CoV-2, what ends up happening is they stop drinking, they don't eat as much, and as a result, their weight is just plateaued. And so they don't lose weight, but they do in fact show signs of morbidity. And we use this as a good indicator to essentially tell us how they're feeling. And not surprisingly, their lack of weight gain is probably due to the fact that they are suffering some aspects of disease. So this is staining of the nucleoprotein of that virus. So this is a subgenomic, one of those subgenomic pieces where the polymerase has to jump. So you can almost be assured that when it's making this protein, it's also making double-stranded RNA. And I hope what you can appreciate is that, like, by day 4, the airway space, this is where you exchange your oxygen and carbon dioxide, right in this airway here of your lung, it is completely covered in virus. And so you can certainly understand why people are going to the hospital and are having trouble breathing and getting in enough oxygen, because certainly if your airway space is completely infected and these cells are dying or are inflamed, that exchange is going to be compromised. However, as could be predicted by the titers in hamsters, you can see that after day 4, where we have peak virus replication, By day 6, it starts becoming more diffuse. Again, by day 8, again, more diffuse. And by day 14, really interestingly, these animals have now recovered, but there is still virus remaining. And I don't have time to talk about this today, but I would love to maybe get into the discussion about long-term consequences of even those who have recovered from SARS-CoV-2. In addition to this, we see infiltration. So this is just H&E staining of just one example of one lung. This is what it's supposed to look like. Lots of space for oxygen and carbon dioxide exchange. But of course, here we have complete infiltration of all these different immune cells because those chemokines are on so high all the time. If we look to T cells, so this is one of those effector cells that we see coming to the rescue. Remember, this is the call for reinforcements. And I hope what you can appreciate here, this is just in the blood. In a control animal, we see 54% of these cells are in the blood, and you see that this percentage drops. And that's because they're leaving the blood and they're going into the tissue. So you can see them going into the spleen. This is going to be helpful for antibody development. They're going into the lung where the infection is. And of course, they're going into the lymph node where they can present antigen to B cells. And not surprisingly, we did see viral clearance, and this is presumably because we also see the production of spike neutralizing antibodies. So we do see by day 7 already the formation of some antibody titers in these animals that just continues to rise at day 14 and day 21. So these animals probably represent what your average, say, 25-year-old would look like in contracting this disease. So if we go to the lung, so again, we just look at this longitudinal study at a low MOI and a high MOI. And so here in green, you can see the virus rises and then falls, whereas at a high MOI, it starts incredibly high and it just falls the entire time. One of the things that I wanted to have the audience appreciate was that if you look at just those genes that are represented by the interferon stimulated family, so this is that call to arms I was talking about, even when you do see evidence of this being activated in vivo, you can appreciate that you really are only getting half the response that you're supposed to get, and that this response essentially follows the virus level, and it lags behind by about a day. And this is going to be important in the discussion later, but really it's because this response is not coming from the directly infected cells, but from the bystander cells, and probably as a result of much of the debris that's left behind by this virus, including that double-stranded RNA we spoke about. And so you look at different tissues, it's quite interesting what you see. So initially you see the highest viral titers in the nasal turbinate. So in the upper respiratory tract, you have very high virus reads. And then what ends up happening is that because this virus is making such a mess with all of this double-stranded RNA, that call to arms, although compromised, is still being induced to some extent in the upper respiratory tract. And so basically the virus goes further into the respiratory tract to get away from that response. And so as a result, you see that the virus titers in the upper respiratory tract begin to fall at day 4. But at that same time, you see them climb in the lower respiratory tract. So the virus is migrating down further into the lungs. And then again, you see that the lungs become inflamed. So you remember on day 4, this is where we saw all of that nucleocapsid staining. They begin to fall as the virus clears, presumably because we're starting to see some antibodies come in. We're starting to see some of that inflammation come in. But then you see the virus peak in the olfactory bulb. So it's kind of moving up into the brain. And this is probably the cause of losing your sense of smell, for example. And before everything just flatlines. And what I really want you to appreciate is this virus, while the virus is largely contained in the respiratory tract, it induces systemic inflammation. So this is just a heat map. All the red genes are genes involved in either the call to arms or the call for reinforcements. And what I want you to appreciate is that even though the virus is largely contained in the lung and the trachea, this is the same low MOI kinetics and the high MOI kinetics, I hope what you can appreciate is that you have the same amount of inflammation everywhere in the body. And this probably speaks a lot to the heterogeneity of the disease, the fact that some people suffer brain fog, some people are having heart palpitations, some people have GI tract symptoms. And it's not really surprising because really it's the systemic inflammation that is being caused by the virus. And so of course, any comorbidities or underlying conditions that may exist would be exacerbated by such a process. And so I'm going to just quickly end the talk to say, like, can you do anything with this? And so, we do test a lot of drugs. I don't really have time to get into that today. What I really want to talk to you today about was just interferon, because it's been in the news quite a bit lately. And what you can see, even in the biological data, is that interferon is effective at containing this virus as long as interferon gets a head start on the virus. And so the question is, if you take a type 1 interferon, can you actually use it as a prophylactic or a therapeutic? Most of the data would suggest that as a therapeutic, it probably doesn't have as much value. You'd have to treat very early. But as a prophylactic, I think it has some potential. So I was saying that this has actually been in the news quite a bit lately. I think it's going to be a discussion point later on. But really what I'd like to pitch, if you will, is that this idea of interferon in and of itself is not great, because as an FDA-approved drug, it's really only used systemically or by sub-Q injection. And interferon itself, of course, is not a great drug to take in that way, because when you talk about flu-like symptoms or virus-like symptoms, those aren't actually caused by the virus itself. They are caused by the call to arms, that production of interferon. So those symptoms are the things that your body is making as a means of inhibiting virus infection. So of course, if you add interferon systemically, you can get terrible body aches, malaise, depression, hair loss. And as a result, most people who are given interferon, it used to be the go-to treatment for hepatitis C virus, for example, are not very concordant with taking it because it makes you feel terrible. And so obviously, if we're going to suggest interferon as a prophylactic, that's not going to work, not only because we don't want to give everybody an IV or sub-Q dose of interferon to prevent virus infection, but if you feel terrible, obviously no one's going to take it. However, an intranasal administration of interferon would be only localized just to the upper respiratory tract. And just to show you how effective this would be, if you take hamsters and administer interferon intranasally to these, so here you can just see a group of 5 hamsters. Here's our control group, so all 5 have a set level of virus infection in their lungs. It looks about 10 to the 6th PFU per ml, so quite a bit of virus in the upper respiratory tract. If you just simply give them interferon prior to infection, you can see that the amount of nucleoprotein that you see plummets many orders of magnitude down, because now the call to arms had a chance to actually work and be established before the virus got there. And this is not only seen by the levels of viral transcripts, but even at the level of PFU. So here again, you see all 5 animals are kind of hovering around this very high level of 10 to the 6th plaque-forming units per ml. But with the administration of interferon, 3 of these animals didn't get infected at all, and 2 of these animals have a 2-order magnitude drop in viral titers, which of course would mean the difference between hospitalization and possibly life. And this isn't just true for hamsters, so there are actually a number of clinical trials going on, but what I really want to stress is that I'm not talking about systemic interferon or sub-Q interferon. I'm really talking about basically an Afrin or nasal mister into the nose of small amounts of interferon as a prophylactic. And there is this great clinical trial that has been described. It's only on medRxiv from what I understand right now. But basically, it was a hospital where they gave workers a small nasal spray that would put interferon alpha into their nose. And they did it twice a day, and they gave it to 4,000 workers over the course of a month. And what they found was that in that hospital, amongst those 4,000 workers, not a single one of them contracted SARS-CoV-2 or seroconverted, whereas in the same time at the neighboring hospital, they had over 2,000 cases. So even though this isn't the controlled clinical trial we might all like to see, the numbers are there to adequately support this idea that intranasal interferon might prove to be a great prophylactic, not just for SARS-CoV-2, but probably for many respiratory infections. And so with that, I'm going to conclude my talk just to say that hamsters and ferrets and people really suggest that the disease that we call COVID-19 that's caused by SARS-CoV-2 is really the product of this bizarre biology where the virus blocks interferon, which again is not so unusual, but leaves chemokines unchecked. And that part is really unusual. And that combination of those two things sets up a stage where you bring in a lot of adaptive and inflammatory cells and proteins in an environment where you're spewing out a lot of double-stranded RNA and really inflammatory molecules. And the second point I just wanted to make was that, I do think that while it's not an FDA-approved modality yet, the use of intranasal interferon should be considered both for prophylactic treatment of SARS-CoV-2, but maybe also for respiratory pathogens for any future pandemic that might come along. And so with that, I'd be remiss if I didn't thank my people. I don't pick up a pipette anymore, and my group has been tremendous. They have been working around the clock for almost a year now on this. Nobody was working on SARS-CoV-2 prior to February, and now everybody seems to be a near coronavirus expert, and I thank them all for it and all of their hard work. And with that, I'm happy to end the talk here and thank the funding sources, all my people, the collaborators, and if anybody would like the sequencing data, we try to post as much of it as we can onto Geo or into bioRxiv on a paper, or simply contact me. My email address is there. So thank you very much, and I'll stop there.
Thanks a lot, Ben. Wonderful presentation. There's obviously a lot of questions. I can see many of them come in both YouTube and here. We'll take some of those questions later, but now I would like to switch to Florian. And Florian, the floor is yours.
Thank you very much. Let me try to share my screen here. So I was asked to give an overview of vaccines and therapeutics that are in development for our SARS coronavirus 2. And I'll start with vaccines. Of course, if we want to talk about vaccines, we need to talk about immunity and immune responses. And so we have learned a lot about the immune responses, adaptive immune responses to natural infection with SARS-CoV-2 since the beginning of this pandemic. You can see here a model of the virion, and there's two main antigenic targets that you can see here. We have the nucleoprotein on the inside. We know that we get a very good antibody response against the nucleoprotein, but we actually don't know what that means. Those antibodies are not neutralizing. And then we have the spike protein on the surface of the virus, which the virus uses to attach to the cellular receptor human ACE2. This is actually a humongous glycoprotein, much bigger than, for example, the influenza virus hemagglutinin. It's a homotrimer. And we know that antibodies against this spike protein can neutralize the virus in vitro, specifically if they are directed against the receptor binding domain, which is this little piece of the spike that binds to ACE2. We have also learned now in the last few months that we get relatively strong T-cell responses against the virus. Usually those are focused, or those are stronger in the CD4 compartment, and the CD8 responses are relatively weak. So that's what we know so far about adaptive immune responses. Now, in animal models, we were also able to look at protection from reinfection. And I'm showing here two studies from Dan Baruch's lab that came out earlier this year. On the left side, you see what happens if you infect non-human primates, in this case rhesus macaques, with SARS-CoV-2. You see here replication in the lower respiratory tract. You get very nice replication over the course of 7 to 21 days. And then if you wait for a few weeks and you try to reinfect these animals, you basically can't. So they are protected from reinfection. Now, of course, this was after a short period of time, and we don't know yet if that holds true in humans. There are reports of reinfections in humans, but usually those are special cases and there's very few reports. The same group also did a vaccination experiment in rhesus macaques with a DNA vaccine, which was just an experimental vaccine. It wasn't that great, and it induced various titers of neutralizing antibodies in the different animals. And then when they were challenged, it was possible to do a correlation analysis between protection and neutralizing antibodies. And what was found there is that there is a very nice correlation between neutralizing antibodies and protection of the animals from infection. They did not find a correlation with CD8 T cells. So in a way, for the rhesus macaque model or for non-human primates, we already have a correlate of protection from SARS coronavirus 2 infection. And that, in this case, is neutralizing antibodies. So, you know, finding things, immune responses that correlate with protection is very important. It helps us with vaccine development. It helps us with finding out who's immune and who is still susceptible. And historically, we have done a very good job with that. There's many correlates of protection for many different viruses and vaccines. I'm just going to point a few out here: hepatitis A and B, influenza. We know if you get an HI titer of 1 to 40, our risk of infection is going down 50%. We have the same for measles. And so the idea is that the neutralizing antibodies might be something similar for SARS coronavirus 2, and we might be able to use them as correlates. Now, so far, there's very little data in humans that suggests that neutralizing antibodies are a correlate of protection, not because they might not be one, but just because there's not enough studies. There's one study that was done by investigators in Seattle that has some promising results, but it's based on a very small number of individuals. In this case, commercial fishing vessel was basically the whole crew was enrolled in a research study before the ship left to sea. And there were 122 individuals on board. Only 3 of them had neutralizing antibodies when the ship left. And then, as you know, ships are a very good environment for outbreaks with viruses. They had an outbreak on this fishing vessel, and they had a very high attack rate because, of course, there's a lot of interactions and a lot of crowding, and the attack rate was 82.5%. But none of the 3 individuals that had neutralizing antibodies got infected. And while these numbers are small, they are statistically significant. And I think that's kind of a glimpse of hope that neutralizing antibodies will protect us from future infections with SARS-CoV-2. Before we go into the vaccines, I also wanted to talk a little bit about what we want to get protected from, and that's also to manage expectations. We know that the lower respiratory tract in humans, if you think about antibodies, is protected by IgG, and that IgG actually correlates with IgG levels in plasma or in serum. And you can induce these IgG by intramuscular vaccination. Protection of the upper respiratory tract correlates more with mucosal immunity, and what's prevalent on the upper respiratory tract is secretory IgA. And this is something that you cannot induce by intramuscular vaccination. And so if you look at the vaccines that are in development, they're all injected vaccines. And so I think what we should expect is that if these vaccines are successful, they will protect us from disease and from protection of the lower, from infection of the lower respiratory tract, but they might actually not protect us from getting infected. And that's something that needs to be kept in mind. All right, so for those of you who know in vaccine development, who work in vaccine development, I know you share my pain when looking at this slide. Vaccine development is a very long process. You start with, exploratory experiments, design of antigens, iterative testing of antigens in animal models. Once you have good results, you might find a commercial partner that wants to take the vaccine on. Then you start a more formal process, formal preclinical experiments, toxicology studies. This might take 2 to 4 years. You also need to develop a process. Then you go into phase 1, which usually takes 2 years. This is followed by phase 2, which might take 2 years. And if the results are really, really promising, and if there is still a market for your vaccine, you might go in a phase 3 trial, which is extremely expensive and takes 2 to 3 years. If that is successful, if you meet the pre-specified criteria, you're going to submit the biologics license application, and then the FDA or EMA or your regulatory, local regulatory body probably has some questions, and the process towards licensure also takes 1 to 2 years. And then you start to produce the vaccine. So it's about a 15-year process. Now, for SARS-CoV-2, this was basically contracted to 10 months to maybe 1.5 years. The design and exploratory preclinical phase was completely skipped. We knew that the spike protein was the target. There were designs for stabilized spikes that had been developed with SARS-CoV-1 and MERS. And so those were the antigens of choice that were moved forward right away. In terms of process development, what happened was that processes from other vaccines were just used for making SARS-CoV-2 vaccines. So a lot of time was saved there as well. And very often the animal data that's needed for an IND was also used from other vaccine studies. And then when we enter the clinical phase, what you see a lot is that the clinical phases are overlapping. Usually the first phase is not a phase 1, but a phase 1/2 trial. And then after an interim analysis, phase 3 trials are started. And at the same time, production of the vaccine starts at risk. Nobody in the industry who is sane would do that outside the setting of a pandemic, because this costs a lot of money. And if the phase 3 doesn't work out, you might have to throw out all your vaccines, and all the money is wasted. And now with a lot of phase 3 trials actually going into already a late phase, we'll see how it works out with getting regulatory review and regulatory approval. There are certainly plans of vaccine companies to get an emergency use authorization first before they get the regular licensure. So the process is really cut down to months, and it's expected that we'll see vaccines with an emergency use authorization, maybe even still in 2020. So now in terms of vaccine candidates, what I'm showing you here in the middle is again the virus itself with the spike protein, and there's many different vaccine candidates in development, and many different platforms are used. The most traditional one is to basically grow up the virus in viral cell culture, for example, inactivated with chemicals, beta-propiolactone. This is what's shown here, and then use that as vaccine. And there are several companies in China, also Bharat in India, a company in Kazakhstan, and now also in Europe. They are developing vaccines like that. It's a very traditional way of doing it and might be successful. You can also attenuate the virus. This is a little bit more complicated because typically the old way of attenuating viruses takes a long time, and reverse genetics with SARS-CoV-2 is also not so easy. There are candidates at the preclinical stage, including one that's developed by Codagenics and Serum Institute of India, but they are not in the clinic yet. Then we have viral vectors, basically replication-competent virus vectors and replication-incompetent virus vectors. In terms of replication-competent virus vectors, the candidate that's furthest along is a candidate that's based on a measles vaccine strain that is expressing the SARS-CoV-2 spike, and that's developed by Merck. In terms of the non-replication competent vectors, we of course have many candidates, including the CanSino Ad5, Janssen, AstraZeneca, and so on and so forth. So those are very far in clinical trials. You can also take a viral vector that displays the spike on the surface and then inactivate that. And that's a strategy that's also developed by Bharat and Jefferson University in the US. You can use recombinant proteins, either the whole spike or the receptor binding domain. And these approaches are also in clinical trials, with the whole spike domain being in phase 3. That's a trial by Novavax. And then we have virus like particle vaccines, which are also in clinical stage development already. We have DNA vaccines, basically just a plasmid that expresses the spike protein under mammalian promoter. There are some trials in phase 1 and phase 2, but they haven't advanced much lately. And then we have the messenger RNA vaccines, where you have an mRNA that's in vitro transcribed and that's packaged into lipid nanoparticles. And of course, those are the vaccines that are developed by Moderna and by Pfizer. I wanted to show this slide because it really shows you the effort that's going into developing these vaccines. We have an astonishing number of vaccines that are in the preclinical phase. I checked today on the WHO homepage, it's 154 officially right now. We have a large number in clinical trials. I think it's 44. And of those, 10 are already in phase 3 trials. So this figure here is outdated. It's a few weeks old. We have 10 phase 3 trials ongoing right now, or 10 vaccines in phase 3. It's many more phase 3 trials, actually. And we even have licensed vaccines. There are asterisks there because these vaccines have been licensed after phase 2. One is the Russian vaccine that was developed by Gamaleya Institute that was licensed after phase 2. The second one is CanSino's vaccine, which is licensed for use in the Chinese military. Now, what do we know about these vaccines? These are data from non-human primates where animals were vaccinated and then challenged. It's a lot of data. I'm not going to go through all of it. I just wanted to point out a few things. We have these inactivated vaccines, which are developed mostly in China. They have been given to non-human primates once, twice, 2 or 3 times. So it's usually prime boost or prime boost regimen. What we see there is that you get okay neutralizing antibody titers. They're not that great. And what you see is if you challenge these animals, you get complete protection of the lower respiratory tract, but you still see virus replication in the upper respiratory tract. AstraZeneca, and that's the Oxford vaccine, also used the non-human primate model to look at their vaccine effectiveness. They gave their vaccine either once or twice. If they give it twice, they get okay intermediate neutralizing antibody titers. And then if they challenge, they see protection of the lower respiratory tract, but again, no protection of the upper respiratory tract. Janssen is special because they used a one-shot vaccination regimen. This vaccine is based on an At26 vector. They get intermediate range neutralizing antibody titers. And what they saw was complete protection in the lower and the upper respiratory tract, but they used the lower challenge dose. Moderna also had encouraging data. They actually get very high neutralizing antibody titers after 2 shots of their mRNA vaccine in non-human primates. They see, again, protection of the lower respiratory tract, but not necessarily of the upper respiratory tract. And finally, Novavax used a recombinant protein vaccine, spike protein expressed in insect cells. They get sky-high neutralizing antibody titers if they give it twice with an adjuvant. And they saw complete protection of the lower respiratory tract and almost complete protection of the upper respiratory tract, but they challenged with a much lower dose. So take-home message, it's easy in non-human primates to protect from disease, but it's not so easy to protect from infection. If you look at the available human data, the inactivated vaccines, when given to humans twice or even 3 times, gave intermediate neutralizing antibody titers. If you look at the vector vaccines, CanSino, which is an Ad5 vaccine, didn't fare that well. You got a T-cell response, but very low neutralizing antibody titers. The AstraZeneca vaccine gives intermediate range neutralizing antibody titers after 2 shots, but also after 1 shot. It looks pretty good after 1 shot too. And then we have the 2 mRNA vaccines. The take-home message here is that after 1 shot, not much happens, but if you give these vaccines twice, you get relatively high neutralizing antibody titers. And again, Novavax did the best in humans so far with the adjuvanted vaccine. If you give that twice, you get titers, neutralizing antibody titers up to 1 to 3,900, which is really fantastic titers. So just to sum this up, vaccines in phase 3, we have 2 mRNA vaccines, one by Moderna, one by Pfizer. They are doing well. They give you nice neutralizing antibody titers. We have AstraZeneca, seems to do well as well. It was on hold briefly in the UK. It's still on hold in the US, but I've heard that the FDA will lift that hold soon. Janssen started in the US and this was on hold relatively quickly. So we don't know what's going on there, but I want to point out that an At26 vectored vaccine is actually licensed in Europe. That's for Ebola. So that made it through phase 3 trials without major issues. So it might not be a big issue that we are looking at here. We have Novavax. Novavax is in phase 3 in the UK. We have the Gamaleya vaccine, which is the Russian vaccine, where an adenovirus 5 is given, followed by an At26, or the other way around, I always forget. This is now in phase 3, but it's also licensed, which is kind of weird. We have the inactivated vaccines. There are 3 very far in development. There are rumors that the Sinopharm vaccine will be licensed in China relatively quickly. And then we have CanSino. Again, this is an Ad5 that is also far in development. And then we have another vaccine that was a few days ago announced to be licensed in Russia, which is called Epivac Corona. That's based on peptide. So I'm not sure what I think about that one. Again, there's no phase 3 data for that. A few words about reactogenicity. We see that there is quite some reactogenicity with some of these vaccines, mostly either local or systemic, but usually transient, so not necessarily dangerous, but unpleasant. And what we see is that for the adenovirus vectors, they are the worst in terms of reactogenicity, followed by mRNA, followed by the recombinant protein vaccines, and the inactivated vaccines actually have the best reactogenicity profiles. What is interesting is that for the mRNA vaccine, the first shot seems to be relatively okay, but when you get the vaccine the second time, the side effects really go up, including fever in a large proportion of study participants. The other thing that I wanted to point out, because it has now been seen for several of these vaccines, independently of the platform, is that the vaccine works much better in younger people than it works in older people. And this is shown here. This is from the Pfizer data. BT162B2 here is the one that is the candidate that's moving forward in the 30 microgram dose. And you see that in younger adults, we get neutralizing titers of 1 to 300, above 1 to 300. Well, we do not get to these levels in older people. It's about 40% of what the young people get. And this has also been seen by CanSino, and it has been seen by Sinovac. And that's concerning because it's the old people that we need to protect. The young people are better off anyways. So we'll see how that works out. A couple of issues to also keep in mind. If vaccines are licensed and they enter the market, that doesn't mean that they will be available to everybody right away. It's probably first very few doses. There might be issues with large-scale production, specifically with technologies and companies that have never licensed a vaccine before, like Moderna or Novavax. They don't have vaccines on the market. But also Moderna, also Pfizer might not have the experience how to make RNA vaccines in the billions of dose range. Distribution might be an issue with the number of vaccines needed. The same is true for administration. And then you have to keep in mind that it takes some time until immunity sets in. Most of these vaccines have to be given twice, and it probably from the time you get the prime vaccination until you might be protected, it's probably 2 months. So in conclusion, for the vaccine part, several candidates induce strong neutralizing antibody responses in non-human primates and in humans. That's great. We do not know if these neutralizing antibodies protect humans, but we should figure that out relatively quickly. We see in the monkey model very good protection of the lower respiratory tract, indicating protection from disease, but we still see infection of the upper respiratory tract, and that might mean that you might still get infected and you might still be able to pass on the virus even if you're vaccinated. And right now it's unclear how the vaccine induced responses actually compare to what we see of the natural infection in terms of protection and longevity. Longevity is also an issue that we need to follow up on. Of course, we don't know yet how long immunity will last, but it might be that booster doses after 1, 2, or 3 years might be needed. That's not a disaster, but it's something to keep an eye on. And now I wanted to also briefly talk about therapeutics. That's not my main area of expertise, but I know a little bit about it. If you look at therapeutics, we have 2 types that are in development and getting tested right now. We have antivirals, which basically can act early in the infection and act against the virus. And we have immunomodulatory drugs that usually work better late in the infection, at least in some cases. And 2 of the ones that I list here are actually meant to mitigate an overwhelming immune response that might do the damage late in the infection. For antivirals, we have monoclonal antibodies. You've heard about the Regeneron mix, which was given to Donald Trump. And then we have one that is developed by Eli Lilly. The Eli Lilly one is actually on clinical hold, so there were issues with that. The Regeneron just filed an emergency use authorization for their monoclonal antibodies, suggesting that they have some efficacy in humans. And those antibodies work in animal models. There's also data on convalescent plasma. I'm not going to talk too much about that. There are still controversies about that, but it certainly works in animal models. We have remdesivir, which is a small molecule nucleoside analog, and that works in animal models very well too. And then we have hydroxychloroquine, which has been shown not to work in humans and not to work in animal models. And lopinavir and ritonavir, which barely work in animal models, and as you will see, have been shown to not work in humans. And for the immunomodulatory drugs, we have interferon. Different than what Ben was talking about, we have IL-6 antagonists like tocilizumab, and then we have dexamethasone. And I'm just going to show you 2 or 3 studies that recently came out and might be of interest. The first one is with remdesivir. This was licensed by the FDA yesterday, so that's not an EUA, that's an actual license. And it has been shown in a trial to reduce time to recovery from 15 to 10 days. And there was a trend towards lower mortality, but the trend was not statistically significant. Now, also recently, the results from the Solidarity trial that the WHO was leading came out on medRxiv, and that included trials in 30 countries with more than 10,000 participants for remdesivir, hydroxychloroquine, lopinavir, and interferon. This was injected interferon. And what they saw is that basically none of these drugs have any influence on the mortality outcome, which is not necessarily with remdesivir a different result than the other trial found. But of course, it's also pretty disappointing, because that means that none of these drugs are helpful in reducing mortality. Then there is also a success story with dexamethasone, which is a corticosteroid, and that was trialed in the UK. And what was found there is that specifically in the later stage of disease, for example, when you were already on invasive or mechanical ventilation, that actually reduces your risk of dying. So this was a large study, and that seems to have a significant effect in the late stage of disease. And another drug that was initially thought to be helpful is tocilizumab, which targets interleukin-6. And a lot of people were dosed with that antibody, with that monoclonal antibody initially. And there was a study that was just published that showed that there's actually no effect either. So that doesn't really improve the outcome of patients. There's actually another paper out there suggesting that giving tocilizumab might increase the chance of getting a secondary bacterial infection. So the conclusions with the therapeutics are, less positive than what we see with the vaccines. We certainly have remdesivir, we have dexamethasone, potentially monoclonal antibodies and plasma, and those might have a positive effect. But the problem is that the effect size is small, and so far we really don't have a silver bullet. And the other problem is, even if monoclonal antibodies are found in the end to be very effective, it might still be an issue to make enough of them and for reasonable costs. So we really need new drugs to deal with the virus during the early stages of infection, and B, to deal with the immune system during the late stage of infection. So far, we really don't have anything that works very well. And with that, I just wanted to thank my team and my collaborators at Mount Sinai and elsewhere. Thanks.
Thank you for that very comprehensive summary of both vaccines and therapeutics, Florian. That was wonderful. I would like to initiate a panel discussion and welcome our panelists, Professor Gagandeep Kang and Professor C.S. Pramesh. Ben, I would like to touch upon something that you did bring up. The potential, the interferon therapy was extremely promising at one point. And now this trial suggests that it doesn't seem to have any effect, at least in hospitalized cases. There's this idea of if an early intervention could be made, this may still be useful. Would you like to comment on that?
Yeah, no, sure. In fact, I think it makes a lot of sense that interferon might actually be damaging latent infection, and there is some data to suggest that, and even the curves that Florian just showed, if you look at them, actually the mortality rate is a little bit higher in response to interferon, and that's, because, as I said, the systemic administration of interferon, whether it's sub-Q or IV, it does do a lot of things. It activates a lot of immune cells. It induces malaise and joint pains and headaches, and it gives you flu-like symptoms. And so, for a patient who is already on a respirator or hospitalized and, is clearly sick, to exasperate that by adding in interferon perhaps isn't surprising that doesn't help, especially because if you look at those patients, like I said, there is an interferon response. It just doesn't come from the infected cells. So it's not effective at stopping viral load. It's essentially pushing the virus into new tissues, into new areas, and it's trailing behind the virus. So there is interferon there already. So adding more is not necessarily going to be helpful. And in fact, it's been shown to be, if anything, the exact opposite. However, what I'm talking about is one, localized, so you'd want to keep it in the upper respiratory tract, but also use prophylactically. So you increase basically you warn the cells that a virus is coming each and every day until, while you're in a high risk environment. And in that sense, I do think it would be very effective prophylactically. It would probably be also effective if you could actually spot, people with comorbidities or of advanced age who were sick but not quite yet hospitalized. If you could identify them and give them interferon early, that might also prove beneficial. And there's work from Eleanor Fish to suggest that is true. So it really is more about timing. However, finding people early because it's so challenging, I think the real value there is mostly in the wheelhouse of being prophylactic only.
Thanks. I would like to bring in Dr. Pramesh, we started talking about the Solidarity trial. There's now a few trials, Solidarity, Recovery, etc. These are large international collaborative trials. This is not something that has been attempted at this scale earlier. Wondering if you wanted to talk about these, what are the advantages of these? Do we gain a lot by doing these kind of large trials over, a lot of dispersed small trials?
Thank you, Aurnab. And let me start with a disclaimer that I'm a big fan of these large pragmatic trials, because everything that we've seen conclusively as part of the COVID-19 response and the treatment options with COVID-19 have come as a result of these large pragmatic trials. You need two ingredients for trials like this to succeed. The first is to have the platform ready, which means that you can't invent a new multi-arm, multi-stage trial design and suddenly try and plug in a pandemic situation and the multiple challenges thereof. And I think it's thanks to the MRC who kind of pioneered this as part of their several other MAMS trials, for example, the Stampede trial for prostate cancer, which enabled a trial such as Recovery and, for that matter, Solidarity to be rolled out in pretty short time. The second aspect of these large pragmatic trials is the fact that they mimic the real world situation far more than the typical phase 3 pharmaceutical funded sponsored trial. So instead of having a whole lot of eligibility criteria which make the generalizability of a phase 3 trial fairly questionable, the advantage with these kind of trials is that it brings about immediate generalizability. So if you look at the Recovery trial, for example, most patients who fit the criteria, eligibility criteria, actually got recruited into the trial. And that's a big advantage, because you know that the results of this trial are likely to be replicated across the board when you implement it based on the positive or the negative results. And finally, to me, the most encouraging part has been the immense collaboration that's happened in a very short time with both these trials. It didn't work as well with the Discovery trial, with the French started, but both with Recovery and with Solidarity, you saw this huge group of researchers across the world breaking silos, breaking boundaries, national, international, and getting together to recruit patients onto what was seen to be clearly important clinical questions. Were these drugs important enough to be tested, and can we get an answer pretty quickly? And if you look at both these trials, they recruited something close to, over 11,000 patients in less than 6 months, which to me is unprecedented in any clinical trial. And just to give you context, pharmaceutical companies struggle outside of a pandemic to recruit a few hundred patients a year. And here you have the other extreme where you recruited over 11,000 patients in less than 6 months and coming up with very important public health questions which are getting answered in a very robust way. Unfortunately, many of the drugs tested in both these trials failed the test. But to me, a negative trial is as important as a positive trial. And at least we laid to rest the questions about drugs like hydroxychloroquine, like lopinavir, to some extent remdesivir. And let's not forget the success story with dexamethasone. So though it was being used pretty popularly across several institutions, we didn't have the level 1 evidence till the recovery trial came on. And to me, the more encouraging part is the fact that the platform is ready to test more promising treatment options. So for example, recovery is still testing on azithromycin, it's still testing on convalescent plasma, and very recently it's even started testing on the monoclonal antibodies from Regeneron. So I think the importance of having a robust public health system and the fact that you have a platform to test these are extremely important. And to date, probably the only things that have mattered in therapeutics in COVID-19 have been thanks to these trials.
Thanks. I want to change gear a little and go to Florian. Florian, the phase 1 and phase 2 results that you presented in humans, a lot of focus is on neutralizing antibodies. Clearly, of course, you're also looking at T-cell responses, but it seems that the assays used to quantify neutralizing antibodies are very different in each of these trials. Could you comment on that? And is there any effort to standardize these so that direct comparisons can be made?
Yeah, no, very good point, and I complain about that actually a lot. So it's hard to directly compare them. In a way, you have to take these comparisons with a grain of salt. Some companies use assays that are based on IC50s or IC80s, IC90s. Some use reporter viruses. Some use CPE as readout. Some have a more quantitative readout. So it's hard to do that. The Oxford group, for example, did 3 different neutralization assays, and you get 3 different results, right? There are efforts to standardize that in a way. There is an international standard for antibodies or for serum to SARS-CoV-2 coming. But I think if you really would like to compare these different vaccines and do a standardized analysis, you would have to have a central laboratory that does it. I think right now, what we will see is a direct comparison in terms of effectiveness or efficacy. But even there, it's hard because the endpoints are not always the same, right? So I think that the real comparison in the end will probably come later when academic researchers and clinical researchers will start to compare those vaccines. So it's not so easy.
So I would like to bring in Professor Kang at this point. And Gagandeep, perhaps you could tell us about what the status of the vaccines being developed in India are.
Sure. So we have currently about 30 vaccine programs in India, and they span every platform that is available in any part of the world. We have 4 vaccines that are in clinical trials, and the first one will be going into phase 3 clinical trials fairly soon. That's the Covaxin, which is the inactivated vaccine made by Bharat Biotech. We also have studies with AstraZeneca's Chadox vaccine that is being tried by Serum in about 1,600 individuals at the moment. And we have Zycov-D, which is in phase 2 and phase 3 is being planned. That's a DNA vaccine made by Zydus Cadila. We have a tie-up with Gamaleya's adeno-vectored vaccines with Dr. Reddy's laboratories, and they have to do early phase testing before they can move to later phase trials. There is a fair amount of work in academic groups and in smaller companies looking at other platforms as well. And one of the things that speaks to what Florian just said is that the Translational Health Science and Technology Institute is going to be one of the laboratories that will participate in the CEPI network for evaluation of vaccines using standardized assays. I also agree that that is a critical component of us being able to understand what the immunogenicity of these vaccines is compared to vaccines in other parts of the world.
Gagandeep, this leads me to another question, a little bit along the lines of what's happening with solidarity and recovery. After the Ebola outbreak, there was an effort to set up a standard platform, the CEPI, the Coalition of Epidemic Preparedness and Innovations. Wanted you to say a little bit about this, and did this effort actually, hasten the production of vaccines for COVID-19?
Disclaimer, I'm vice chair of the board of CEPI, and I was on the scientific advisory committee in the interim phase. So I think CEPI was a game changer. So after Ebola, there was a lot of discussion around the world about what needed to be done in order to have vaccines for emerging infectious diseases. And CEPI was set up with defined targets and disease X, so platform technologies that could be used to rapidly develop vaccines. It's because last year CEPI had funded 6 programs for platform technologies that made it possible to develop, transition all the MERS coronavirus programs to look at SARS-CoV-2. So we had 3 programs that were funded by the end of January. And there are now 9 programs, of which 8 are already in clinical testing. So most of the front runners were funded initially by CEPI. CEPI has tiny amounts of money, but it functions really as a catalyst. So I really think it has been a game changer. I just saw Florian's comment in the chat box, and Florian, I'm very happy to tell you that the US is part of CEPI as of last week. So tiny amount of money, but finally come on board with funding from USAID. Over.
Ben, maybe going back to you, couple of things. What are your thoughts on other strategies that may leverage the innate immune system to fight this disease? And perhaps even if those possibilities are out there, there's the possibility of genetic risks due to mutations damping the interferon response, for example. Is there any evidence for that?
Yeah, that's a lot of question. I guess to be brief, there are huge efforts right now, of which my lab is a part of, to look for repurposing of FDA-approved molecules. This really is not a great option, but it's one of the better ones we have, only because if we could essentially find a side effect of an FDA-approved drug that was designed and marketed for some other disease or attribute, that it might be immediately deployable to the human population. So there are large efforts right now doing high throughput screening that start with basically like the dishes behind me, just cell culture, looking for any drug that might inhibit virus first in cell culture, and then we advance to more advanced cultures like organoids or gene on chip, and then we ultimately try in hamsters. And I do have to say that the vast majority of molecules that work in cell culture ultimately fail in animals. This is actually the story of hydroxychloroquine as well. I have found a few that work. One of the ones that I really like is this derivative of the hydroxychloroquine called Amnio Diaquin. But that drug in and of itself, it's a perfect example of it's not a perfect drug. It's actually quite a dirty drug that has been given a red box delineation from the FDA because it has been associated with some severe diseases. But on the other hand, it's given to 30 million kids in Africa to prevent malaria, and it actually works in hamsters to reduce viral load quite significantly. So, maybe there's hope there, but really that's, we went through this exact same exercise for the Ebola outbreak, and it really didn't come to fruition at all. I also like, though, as far as leveraging the transcriptional response, the work done by a lot of people using artificial intelligence and transcriptional signatures. So there's some great work where you look at the genes that move up and down in a variety of different pathways and annotations. And it doesn't really even matter if you understand the biology behind it, but you start off by saying, you take a respiratory infection in a human, and you say, okay, SARS-CoV-2 induces pathways A, B, C, and D, they all go up, and pathways F, G, and H all go down. Do we know of any drugs that do the exact inverse to those same biologies? And there has been an effort over the past 10 years, it's called the Lynx 1000 project, where they take FDA-approved drugs and they add them to various cultures and sequence them and then make those same annotations. And so you can quickly use computer algorithms or different mathematical algorithms to look for drugs that run antagonist to the pathways that are beneficial for virus. And those have also pulled out some very interesting compounds that are also now being tested in animal models. But really all of these are poor substitutes for an antiviral that would be directed against some enzymatic component of the virus directly. But it's what we have, and we're trying to play catch up essentially. With regards to your very last question, I mean, certainly inborn errors of innate immunity have shown much more severe cases of disease. And again, that's not at all surprising. You can imagine that if you take the small interferon response that we do observe in COVID patients and take that away altogether, of course, the virus runs rampant and is essentially unstoppable. And that's why we see people with TLR7 mutations, showing very high lethality rates, also true for interferon signaling. There's also this report about older men making antibodies to interferon. So, essentially a mistake of our own immune system where we take out the little bit of call to arms that we have, and that also induces more severe disease.
One last question, and I'll ask Florian and maybe Gagandeep to talk about this. There's been a lot of talk about cross-immunity originating from infection by other coronaviruses or even from vaccines, BCG, for example. Your thoughts on that, Florian, and then maybe Gagandeep?
Yeah, so I mean, from other vaccines that are completely unrelated, I think the only thing that you could get is some innate immune response that is upregulated that might give some protection, but I do not believe that is a viable concept. What you see is that there are cross-reactive T cells, both CD8s and CD4s, from human coronavirus infections to SARS-CoV-2. We do not understand the relevance of that. Could be that they contribute to lower disease severity, and that could help to explain the heterogeneity that we see in the severity of infections. But as you know, T cells are unlikely to protect you from an initial infection, right? They kick in later, they remove infected cells. We do see cross-reactive antibody responses in an original antigenic sin fashion. There is now 2 or 3 preprints out there that show that with OC43 and HKU1. But again, the relevance of that is unclear, and these antibodies don't seem to neutralize.
I guess, we are at the end of the time allocated for the panel discussion. So thank you, everyone, for this. I will hand over to Janine and Sarda to handle the question and answer sessions.
Thank you. Thank you, Ben. Thank you, Florian. Thanks to all the panelists and Aurnab. That was a very stimulating discussion session. And now let's move on to questions from the audience. So the first question is for Ben. Ben, so you clearly showed that there is an imbalance in immune response that's underlying this disease, so mainly an imbalance between cytokines and interferons, right? So is this signature unique to SARS-CoV-2? Or is there a difference in this imbalance between SARS-CoV-2 versus SARS-CoV-1 or even MERS? And if so, is that the reason for the debilitating effects of SARS-CoV-2?
That's a good question. There's no comprehensive, simple answer to it. So the antagonists that SARS-CoV-2 have appear to be a little different than SARS-CoV circa 2003. One of the main antagonists, the ORF3b protein, is actually truncated in SARS-CoV-2, and it's unclear whether or not this tiny protein is responsible for only the partial block that we see against IRF3 and maybe the continued production of chemokines. So certainly some small polymorphisms in SARS-CoV-2 may in fact be the result of the high chemokine production in the background of low interferon. So both SARS-CoV-1 and SARS-CoV-2 both block interferon production, but the high chemokine production does seem to be an attribute of SARS-CoV-2. In addition, SARS-CoV-2 ironically seems to be more susceptible to interferon treatment in contrast to SARS-CoV-1. This is work from Vineet Manachari, posted on bioRxiv, and he showed against live virus that really if you perform the same experiment that I discussed where you pre-treat with interferon, that it is an effective way at stopping SARS-CoV-2, but not so much SARS-CoV-1, suggesting that downstream antagonists, so the virus also has this ORF6 protein, so open reading frame 6 protein, that is very good at blocking the nuclear translocation of transcription factors. So the ability of a cell to actually respond to virus demands that you send things into the nucleus to induce a response, and the virus blocks that process. And so it seems likely based on that data, although it has not been formally shown, that SARS-CoV-1 might be better at doing that as well. So those are certainly attributes that are adding to the pathology of SARS-CoV-2, but certainly the new receptor is going to change a lot about the biology, how fast it can get into the cells, how well it transmits, and all of these things, of course, are playing into what has occurred.
Thanks, Ben. The next question from one of our audience members is a two-parter. First is, why are you using the hamster model instead of mice? And also, they noted that relatively low titers of neutralizing IgG serum titers appear sufficient to protect primates and hamsters from lung pathology. Are there any insights into why this might be the case?
All right, so the first question is an easy one. As probably many on the panel know, mice, the ACE2 receptor of mice has a few polymorphisms that prevent the wild type spike protein from binding and engaging that protein. And so you have a couple of models. If you want to use mice, you can either adapt the virus by changing the spike protein to be able to recognize the mouse protein, and the Ralph Barrick lab has now done that. Or you can change the mouse to either express human ACE2, which has been done, or you can actually modify the endogenous locus just to change those few polymorphisms so that they look more human. Every one of those mouse models has pros and cons to it. The disease in mice is more severe. One major problem with at least the transgenic mice expressing ACE2 is that ACE2 is expressed in cells that are not pneumocytes. And so you are getting replication and infection in cells that wouldn't normally be infected. So there is a sense of non-physiological settings there. And so the mouse model certainly has value for testing vaccines, but with regards to looking at the actual biology of the virus, I really think maybe the mouse-adapted virus in mice works, but changing ACE2 expression and modifying it, any leaky expression that you get is going to cause phenotypes that wouldn't normally exist in a real life situation. So the mouse model is a bit limited. The hamster model, it also has limitations. Like no model is perfect. In the hamster model, we lack a lot of reagents. The genome's not even sequenced. I actually found my first gene the other day, so I mapped out the interferon beta gene of hamster. But that just tells you how little we know about the genome of hamsters that we had to go digging for it to go find it. However, the hamster model doesn't require any modification of the animal, doesn't require any modification of the virus, but there it also mimics that males get more sick than females. The older the males get, the more sick they get, and so it really does recapitulate a lot of aspects of the disease. But now you're trading it against the fact that we don't have any reagents or knockouts, and so that's not great. The second question is a difficult one to answer. Why they clear the virus is less clear. Like for example, in the ferret model, we see upper respiratory tract infection, and that virus gets cleared even in the absence of interferon or in the absence of antibodies. So it would suggest that maybe because the virus is less capable of efficiently knocking down some of that innate immunity in hamsters or ferrets, because they're obviously not humans, that might contribute. So maybe the interferon response is a little bit stronger, and so it's a combination of the generation of antibodies and a more robust innate immune response. That could be it. But that same thing might also play out in, say, a 25-year-old, where the initial response to virus infection might be strong enough to keep viral titers low so that even a small adaptive immune response to that virus could be sufficient to clear it.
Interesting. Thank you, Ben. And staying on, continuing with the immune response, could you speak to the long COVID or the long-term effects of the infection? I mean, could the damage because of this imbalanced and overactive immune response be the reason for damage to other organs and hence the lingering additional symptoms?
Yeah, so this is actually kind of a scary aspect of the story, and so I don't want to be sensationalist. So this is very early days in this respect, and most of the knowledge that in this area comes from anecdotal stories that you hear on the news or reports from New York Times or whatever. That people, have brain fog following infection, or malaise, or general lethargy, or like I was saying, heart arrhythmias. There's a number of complications that have been associated with renal failure is another one, with people who have recovered from COVID. Now, of course, anecdotal stories can spread far and wide, and when mathematically you look at the statistics, they might not actually be relevant. But it is true that when we look at hamsters that have recovered, you can see in the pathology of the lung that the pneumocytes are expanded. They become very large, hyperplasia. And in addition to the hyperplasia, you get spaces in between them. And now I'm not a pathologist, but the pathologist that looks at our slides, who did it blindly, was very concerned about day 14 because they have now cleared the virus and the virus looks good in the lungs. But what the pathologists refer to it as precancerous. So usually when you get space between cells, that's not supposed to happen. It's supposed to fill in rapidly. And when there is space, I guess that amongst pathologists is a form of precancer. So that idea is certainly a really scary idea, and we are formally looking at that now. I also have to say that we've looked in the brains of hamsters, and even though you get very low virus in hamsters, if you look at the transcriptional signatures of brains, even 20 days out post-recovery, you see transcriptional signatures that have some elements that mirror that of, say, Alzheimer's and Parkinson's. And so, again, I don't want to be sensationalist. I'm only telling you what the data shows. And we are now actively studying a very large cohort of animals that have recovered to see what long-term effects look like. But a very scary thought is that amongst the millions that have been infected, we might have another massive problem to deal with down the road with either some lung disease from long-term infection or even brain chemistry.
All right, thank you, Ben. And now switching gears to Florian. Florian, here's your first question. I'm actually grouping a bunch of questions here. So, the vaccines currently under trial are mostly intramuscular, and this route evokes IgG that protects the lower respiratory tract. So if these vaccines are effective and do get approved, will the absence of secreted IgA mean that we will only be able to enable partial protection and there will be continued transmission via upper respiratory tract infection?
Yeah, that's a very good question. I'm worried about that, and the non-human primate models show that that might be the case, right? Of course, even if you're not completely protected in the upper respiratory tract, you might shed less virus and you might shed for a shorter period of time. So I can imagine that even if that's the case, there would be less transmission from vaccinated individuals. But it could certainly be that, as is the case with the current pertussis vaccine, for example, or something that we also see with influenza vaccines that are injected, that you can still have infection and that you might still have transmission. So, it depends a little bit what consequences that has. But if you have a very high vaccination rate and most of your susceptible and vulnerable population, older people with immune deficiencies, are covered by vaccination, that might not have big consequences, but it will certainly be a complication in controlling the virus.
Great, thanks, Florian. The next audience question is, considering that many vaccines, including the front runners, may only work to prevent lower respiratory tract infection, and this only occurs in a subset of patients, does this suggest that there will be a longer time before we have a satisfactory phase 3 trial result?
For the initial results that may come out, it might be based on relatively short period of time, and it depends a little bit on the endpoints, right? I know, for example, the Pfizer trial, one of the endpoints is certainly infection, but it's symptomatic infection, right? And so if you get infected, but you don't have symptoms, you would be counted as protected, right? So I don't think that that might have a huge impact on getting results from the phase 3 trials.
All right. Thank you, Florian. Here's one for you. Are you concerned about, should we be concerned about vaccine-enhanced disease?
Yes, we should be concerned about it, but we shouldn't be too worried about it. So for coronaviruses, there is some indication that enhanced disease that could either be based on previous infection or vaccination does exist in some models. We know that from feline coronaviruses, where this happens mostly in laboratory settings, not so much in the field. We have seen complications, and you could call that enhanced disease, in some animal models with some vaccines for SARS-CoV-1. And we have seen that also in some cases for MERS. Not in all cases, and it's not always a classical enhanced disease. For example, in ferret models, there was liver toxicity, but the virus was controlled very well. So right now, of course, because people knew that, there was a lot of focus on that, and everybody was keeping their eyes open, but I was actually trying to reproduce this effect in animal models now for SARS-CoV-2. To my knowledge, there is no data right now suggesting any enhanced disease in animal models for SARS-CoV-2, and we have absolutely no data that would suggest that this happens in humans. So I think we need to keep our eyes open. We need to be very careful in phase 3 and in phase 4 trials, so post-licensure trials. But right now there is not really too much reason to worry about it.
Thanks, Florian. So what, in your opinion, are the real major risks of such a drastic shortening, in the research time and the trial time? And also, is there any evidence for protection by vaccines that are used for other viral infections?
Well, there's not just to tackle the second question first. I don't think there's any evidence that other vaccines protect you from SARS-CoV-2 infections. It needs to be a SARS-CoV-2 vaccine. In terms of the risks, so in terms of safety evaluation, nobody's taking shortcuts. That needs to be clear. The safety evaluations are still the same as they are for traditional development, where there's a lot of risk taking is economically. So, there's a lot of money put at risk into development. And if a vaccine fails, all that money is gone, right? So that's kind of how this was sped up. You have to keep in mind a regular phase 3 trial might take 1, 2 or 3 years. So that's your safety follow-up there. And now we are trying to do that quicker, but the actual phase 3 will end after 2 or 3 years. So we'll still get that safety data. You also have to keep in mind that it's massive numbers that are enrolled into these phase 3 trials. For example, if you look at some of the US trials, we're talking about 30,000 to 45,000 people. If there would be a very common safety signal, you would find that. Of course, long-term issues that might arise, you can only identify over time. But that's the same with regular traditional vaccine development, because a normal phase 3 that goes over 2 or 3 years is also not going to identify issues that might come up 10 years later. So in a way, it's basically the same process. And I actually am happy to see that there's so many people enrolled in those trials, because that actually gives you confidence if there's no safety issue that might hold true than in the general population.
All right. Thank you, Florian. Here's one for you. This is on neutralizing antibody. So the first question is, do different trials use the same assay for neutralizing antibody? If not, how do we compare this between studies? And adding to that, the numbers seem to look different between Moderna and Novavax, and is that concerning?
So I mentioned that earlier, it's really hard to compare these things, these different readouts, because the companies develop their own assays. They're using different readouts, different ways of quantitating neutralization, different assays actually. So you have to take comparisons with a grain of salt. For the CEPI trials, there will be comparisons that can be done, because there might be central analysis of the immune response for some of them, but that's not true for all the vaccines in development, because some companies don't take part in that. So it's hard to really compare. The difference between Novavax and Moderna and Pfizer seems to be real. There's a log difference there. I'm saying it seems to be real because in all three cases, when they did the analysis, they also included an array of sera from convalescent patients. And so you can compare it to that as well. And Novavax really hits the upper portion of convalescent individuals. So it seems that their neutralizing antibodies are higher, but again, you have to take these comparisons with a grain of salt.
I think that is about all the time that we have for questions right now. I think that we did hit a lot of the key audience questions, and I thank everyone for the fantastic questions. So at this point, I really just want to thank everybody for their contribution to the series, especially our speakers today, Ben Tennover and Florian Krammer. They were really, really interesting and great talks. I also want to thank our expert panelists, Gagandeep Kang, CS Pramesh, and Aurnab Ghose for the really stimulating discussion. And of course, thank you to our audience for some great questions, and also for tuning in and joining us each week for these really important talks. Lastly, I also would really like to thank the many people who skillfully planned and executed this series, especially Maria Miram and Abi Aron and their entire team from TNQ Technologies, and also the very talented members of the Janelia AV team, and of course my co-moderator and co-organizer, Sarada Viswanathan. So thank you everybody for all of your contributions to the series, and stay well.